vaccines, they're hard until they're not. And you just have to keep trying it. And suddenly one day, a personalized cancer vaccine will work.
As the SARS-CoV-2 virus swept across the world, scientists needed to move fast to create a safe and effective vaccine. Previously, it's taken many years to develop and roll out new vaccines. But with a global emergency, large amounts of funding and willing trial participants, scientists have been able to offer a range of vaccines in just a year.
Some of the first vaccines to be approved were mRNA vaccines, which use genetic instructions to help the body prevent and fight disease. Professor Anna Blakney uses the term RNAissance because of the huge potential these kinds of vaccines have.
They work really well, they have great safety profiles, it's quite easy to produce them if there's an outbreak or a new pathogen that's discovered. And so I think it's just a total game changer for vaccines.
What is an RNA vaccine and how does it work?
I like to think about this in the context of what is a vaccine and how is an RNA vaccine different? So with any vaccine, we're trying to train your immune system to recognize a pathogen without ever seeing it. So we do this by training it to recognize a protein on the surface of the pathogen. So for COVID or SARS-CoV-2, that's the spike protein on the surface, which I think we've all heard about now. So there's many ways of getting that protein into your body. We can just give you the protein, which is kind of a more traditional way of making vaccines, but instead we can also encode that protein in RNA, give you the RNA, and then the patient's own cells make that protein.
Messenger ribonucleic acid, or mRNA for short, is a single stranded molecule that plays a vital role in human biology.
RNA is found in all of the cells in your body, right? So it's like the stepping stone between DNA, which is, your total genetic code, which contains all the instructions for actions that will happen in your body. And proteins are the actual molecules that carry that out. And so messenger RNA is just the stepping stone in between those.
As implied by the name, mRNA acts as a messenger, carrying genetic code from DNA in a cell's nucleus to ribosomes, the cell's protein-making machinery. It is this process that the vaccine taps into, giving the ribosomes new instructions to carry out. But finding a way to do this proved to be difficult.
Your body is really good at detecting foreign RNA and kind of has evolved to have to do that because there's many viruses that are RNA-based viruses. So it's an uphill battle getting it into cells and to get it translated because you have all these defense mechanisms against it.
One of the big breakthroughs was finding a way to deliver the mRNA message so that it was not rejected by the body's immune system. Dr. Mike Watson has worked for Moderna since 2014, one of the companies to successfully create an mRNA COVID-19 vaccine.
We already knew that RNA had huge potential. What we needed to know was that we could deliver it. So the first really important scientific breakthrough is the delivery. We use something called lipid nanoparticles, little fatty globules that allow the RNA to get through the cell membrane into the cell.
It's kind of just like a fat blob that is able to fuse with the cellular membrane and get the RNA into the cell.
This breakthrough happened a few years before the COVID-19 pandemic. So although the technology is new, it is not untried.
We knew that it worked then in the clinic because of these lipid nanoparticles. And we also knew that in relatively large numbers of subjects, it worked pretty well. So I think it's important to say that RNA hasn't suddenly appeared out of a cupboard from nowhere. It really was on course anyway. So what's happened is an acceleration rather than some kind of magic trick.
This breakthrough unlocked the potential for mRNA vaccines. One of the major advantages of using the body's own protein factory is the speed and infrastructure needed to create the vaccine itself. Unlike traditional vaccine technology, where proteins are made in the lab, a difficult and time-consuming process, mRNA is a simple chemical signal that needs a much smaller setup, making it easier and quicker to produce in large quantities. But once the mRNA is delivered, how does it work as a vaccine against COVID-19? After injection, the lipid coat fuses with a cell, allowing the mRNA to enter the cytoplasm and start coding for the spike protein. Both spike proteins and spike fragments protrude from the cell. When the cell dies, these spike proteins trigger the immune system to provide protection. The immune system contains antigen-presenting cells, which can engulf spike proteins from the dead cell and present the fragments on its surface. Another vital immune cell, a T helper cell, can detect the spike protein fragments, which causes them to activate. Then, when an activated T helper cell reaches a B cell with the spike protein fragment on its surface, it activates the B cell, causing it to produce antibodies. Antibodies are proteins that can bind to the spike protein of the virus. If someone is then exposed to the SARS-CoV-2 virus, antibodies bind to the virus spikes, preventing it infecting any cell. Both Moderna and Pfizer-BioNTech vaccines use this method to fight disease and were some of the first to receive widespread regulatory approval for emergency use. Due to mRNA vaccines being a new type of vaccine, a lot of people have been put off, thinking that they have been rushed through or are experimental, but this is not the case.
Before the pandemic, I think a lot of people don't realize how much research had actually been done on RNA vaccines. But the first time that someone ever really used RNA in the context of a vaccine was in 1990. So it's actually been decades of research. Nobody had done a phase 3 clinical trial before the pandemic, but I think it was kind of just right at that sweet spot where enough research and progress had been made to the point where they could easily pivot and, get RNA vaccines made and designed for COVID and then start these big clinical trials. And then, to all of our pleasure and surprise in some ways, that they have really high efficacy as well, which is really excellent.
We can think of this as an accelerator, a moonshot. So it's allowed us to move forward to close what would have been a sort of 8-year gap to more of a year gap.
To ensure they're safe and effective, all vaccines undergo a strict clinical trial process, from preclinical to phase 1, 2 and finally 3. To speed up the trial process, some of these phases have been run concurrently. Both the Moderna and BioNTech vaccine efficacy has been proven to be above 95% against severe and symptomatic COVID-19. However, their rollout has been hampered in poorer countries by the fact that they need to be stored at a very low temperature.
We're essentially dealing with two components that have quite different requirements for stability: your RNA and your lipid. And so think of it a little bit like making salad dressing. You put your oil and your vinegar together, you shake it up, and it's all good for a few minutes, but if you come back in a couple of hours' time, it has separated again. We don't want that to happen, so we have to take all of the energy out of the system so that it can't separate. And the best way to take energy out of any system is by reducing the temperature. As you know, that reduces all the kinetic energy. Until we can solve that compatibility problem, we're likely still to have to freeze them.
Despite this disadvantage, mRNA technology has the potential to work for other diseases too, in particular for influenza or the flu.
We have to decide in February of each year what strains we're going to put into the flu vaccine so that it's ready in October for distribution. But because RNA vaccines are so much quicker to make, right, we could actually, and people all over the world are constantly surveying what flu is going around. You could imagine in the winter having, actually 3 or 4 different flu vaccines that you could get that would be more appropriate for the strains that are actually circulating to hopefully improve the efficacy of the flu vaccine.
Flu vaccines today currently contain 4 strains of flu. So that's 4 different kinds of RNA. So we want to know that we can put those 4 different kinds of RNA together and it still produces the separate antigens. We want flu vaccines to be stable so that everybody can get them, not just those that have access to freezers. And the holy grail of flu vaccines is to find approaches which don't require updating every year, what we would call a universal flu vaccine. So are there parts of the flu that we can produce with RNA that will give you an immune response that protects you for many years?
As well as influenza, research and development has started for mRNA vaccines for many other diseases, such as Zika, rabies, Nipah, and even HIV and cancer. Moderna announced they are moving into phase 2 trials for an mRNA cancer vaccine after positive results from the phase 1 trial. This therapeutic vaccine is given once a patient already has cancer.
So cancer cells look different to the body. And so what we do is we take a sample of an individual's cancer cells, we sequence them to understand what is different from those cells compared to their normal cells, and then very rapidly produce a vaccine that targets those differences.
Professor Blakney and her team have been working with another kind of mRNA known as self-replicating RNA.
Once it gets into a cell, it's able to make copies of itself. So you get essentially a much higher effective dose of the RNA compared to a normal messenger RNA molecule. So why this is really advantageous is because it allows you to use a much lower dose, which, is generally advantageous, right? You're always trying to minimize the dose so that you minimize the side effects associated with that. But in a pandemic, that's hugely advantageous, right? If we could make 100 times as many vaccines from the same batch size, then we'd be able to make billions of vaccines a lot faster.
It's still very early days for the RNAissance, but both Professor Blakney and Dr. Watson are excited for the future.
What we absolutely do have is a really good tool for attacking those diseases that have been so hard, whether that be HIV or cancer. So that's beyond doubt. Will they work? It's us versus biology, and biology can be a cunning foe. So we'll only know when we know. And I have a rather trite thing that I say about vaccines, but it's true. They're hard until they're not. And you just have to, keep doing it, keep trying it. And suddenly one day, a personalized cancer vaccine will work. Or suddenly one day, an HIV vaccine will work. And then we're all good. I can't predict whether that will be in a year's time or 5 years' time or 10 years' time. But it's a fantastic tool, and it's a better tool than we've had before.
It's finished already, but don't worry, we've got loads more Razor stories for you. Don't forget to like, comment and subscribe, and hit the bell button below for notifications. We'll see you next time.